All right, so my name is Jackie Goordial. I'm a student here at McGill University. And today I'm going to talk about University Valley, which is one of the McMurdo dry valleys in the McMurdo dry valleys, which is classified as a hyper-arid cold desert. There you go. And so I'm going to knock off the acknowledgements as the first thing. This is actually part of a very, very big project with a lot of collaborators, most of whom I'm not mentioning here. These are kind of the collaborators in my immediate vicinity. And I'd like to thank all of them, especially Margarita Mironova, because I'm borrowing some of her figures and data for this presentation. And then my financial support. This is part of a NASA ASTEP-funded project, and I pay for my groceries through the Canadian Astrobiology Training Program. So just to give this talk a little bit of context, I'll tell you what the bigger context of my project. And so I'm part of a team that has designed a prototype Mars drill. And so what they've designed is a drill that can drill 1 meter down into frozen ground. And it can be operated remotely, which are two features that would be very nice for a drill that you're sending to Mars. They're also doing all sorts of science around this, and that's where I come in, the microbiologist. And so they were testing this in the upper dry valleys of the Antarctic. And so why the Antarctic? We've talked about this already. Mars is a really, really cold and dry place. And in fact, it's actually the coldest and driest place we have on Earth. So we're going to hear a lot of talks about the Arctic, and we've heard about coastal Antarctica. But the upper dry valleys and the McMurdo dry valleys are really about the best place we have on Earth to study that. And so it's not quite as cold and dry as Mars is today, but we think it might be comparable to what Mars was in the past. And so we've seen a lot of pictures of the dry valleys already, and I really like this photo that I've stolen from someone else's publication. But I really like it because of this small box here. And what it does is it divides the McMurdo dry valleys into different areas. And the McMurdo dry valleys are not just one homogeneous region. And I want to just kind of point out this gray or gray-brown, I'm not colorblind, this brown area, which is the upper dry valleys, or the stable upland zone of the McMurdo dry valleys. And this part of the dry valleys differs from the rest of the dry valleys because this particular area is sublimation dominated. So water in this area either exists as ice or it directly sublimates into vapor. And this is in contrast to the other areas, which do get a bit of precipitation. They do get some thawing and a bit of meltwater. But in this area, sublimation processes dominate. And what happens is you end up getting a layer of dry soil where the water sublimates away. And then underneath that, the ground ice gets stable. And so it's the only place on Earth where you actually get a layer of dry permafrost overlaying ice-cemented permafrost, which is similar to what Brandon was talking about yesterday when he was talking about the Arctic. So the upper dry valleys are a unique analog because they are the only place that has dry permafrost overlaying ice-cemented permafrost. Air temperatures in some of the upper dry valleys never rise above 0, and water exists as ice or vapor. And we've seen this photo already with Phoenix's little scoop and the ground ice underneath it. And just to kind of hammer this one point home, so we have an active layer in permafrost, or associated with permafrost, which is a layer that seasonally goes above 0 degrees. And permafrost is defined as ground. It's technically defined as ground that doesn't rise above 0 every 2 years, but we'll just say it's permanently frozen for our purposes. And in a lot of the upper dry valleys and on Mars, you get this active layer, which in some places of the dry valleys can be as little as 5 centimeters or cannot exist at all. And then you get this dry permafrost layer that overlays your ice-cemented permafrost. And what does that look like? So if you're on the ground, it literally just looks like a desert soil. So this is maybe about a day that's maybe about -20. You can literally just pick it up, and it's just dry soil. So life in the McMurdo dry valleys. For a really long time, the McMurdo dry valleys were just kind of presumed to be dead. There's no plant life. Birds don't fly into it. You can't really see a lot of bacterial life even, except for in endolithic communities. There were some early publications by Horowitz in 1972 where he concluded that the dry valleys were essentially sterile. And this was because they did it based on culture-based studies. Now we have a lot more molecular techniques. But in a line from his papers, the dry valleys are essentially abiotic areas. The microbial population of the region is probably in balance between influx from the atmosphere and mortality on the ground. And essentially what he's saying is a bunch of bugs are just flying in there, and that's what we're seeing. Nothing's actually living there. So The upper dry valley permafrost. What's this like if you're from the microbial point of view? Well, it's really dry. It's oligotrophic. There's not a lot of nutrients here. It's cold. You're getting a lot of radiation, background radiation, and there's a high physical disturbance. There's a lot of winds. There's strong katabatic winds that are kicking up the soil. And so going back to this map, the region that I'm going to be talking about is right there on that red arrow. This is University Valley, and it is one of those in that sublimation-dominated area of the Antarctic. So this is what University Valley looks like on the ground. There's a very old receding glacier at the head of the valley. The valley is about a kilometer in length and half a kilometer in width. And temperatures in University Valley, air temperatures never go above 0 degrees. So this is weather station data for a year. And as you can see, at 20 centimeters depth into the permafrost, for the majority of the year, it's actually quite cold. Some lows to minus 45 degrees Celsius. And about the only place that we ever see it ever go above 0 degrees, I don't know how to, there we go, above 0 degrees is in the summer times, and that's at the surface temperatures, and that's when the sun is heating up the soil surface. Okay, and if you'll appreciate my MS Paint skills, and we have X-ray vision. So this is just a cross-section of that valley, and so these are to represent the cores. These are the samples that I've been working with. That's the glacier at the head of the valley. And this valley is actually really, really neat because as a function of distance from this glacier, the dry soil permafrost actually has a gradient in terms of depth from the surface to depth to the permafrost. We're not actually sure why this happens or how this was actually formed in the first place, but it's a really neat place to test out some hypotheses because if we're looking for liquid water, ice is a potential source of liquid water. And the closer this available liquid water is to the surface, the more likely we may have liquid water at this dry soil ice-cemented ground interface. And so we do have temperature data from here and over here. And even though this is closer to the surface, this actually never rises above 0 degrees. And so I think it's sort of like a freezer. It's essentially sitting on a block of ice. Whereas over here, this is where we get that maximum of 8 degrees Celsius. So the sun is able to heat this up, except unfortunately, it doesn't, this area here never rises to above 0 degrees, even though this gets to 8 degrees Celsius. And so what does this mean for life? It means that in the permafrost, it gets colder with depth, and we know that life needs water. The colder it is, the harder it is for water to exist. Another point I wanted to kind of drop on is we're actually dealing with really, really old sediment here, or soil, sorry. So it ages about 2,500 years with every centimeter that you go down. So this is a really, really great place to look for preserved biomolecules. Just in terms of some direct cell counts, on average, I'm seeing 10 to the 3 cells per gram. This is very, very low biomass. Neutral pH, very little nutrients, and most of my samples is actually undetectable nitrogen, organic nitrogen, and organic carbon. So if you're a microbiologist, there's some common questions that you're going to ask. You're going to ask, who's there? What bugs are actually there? Where is the source? Where did these bugs come from? And an important question to ask is activity. Are these microbes actually live? Do they represent just woolly mammoths? Did they just get deposited in the atmosphere and get frozen into the ground? Or are they actually doing something in this environment? And another thing that you could look for are signs of life. Because we're dealing with really old soil that's very good for preservation, this is a good place to look for biomolecules. And we're doing really, really interesting research in this, but I'm not going to talk about it today. So you can ask me later if that's something you're interested in. So one of the things microbiologists do is they try to culture bacteria. And Jen already mentioned this morning that 99.9% of bacteria are not actually culturable, but we spend many months of our lives doing it anyway. And out of these soils, I've only gotten 6 isolates to date. And that's out of maybe 8 months of trying very hard to get more isolates. 3 of them I would say are particularly interesting, and they're interesting because they grow at very low temperatures. So one Methylobacterium I have that grows at -10, another Rhodococcus that grows at -10, and I just discovered that a Sphingomonas grows at -5 because I left the culture at -5 and then checked on it a year later, and it's growing. But these are also particularly interesting because they're salt tolerant. And this is a trait that you're going to see with permafrost bacteria in general. And I know Jen's going to be talking about that a little bit later and elaborating on that more. So besides the culturing, to get a bit more of a holistic view of the bugs that are in there, we can do 16S pyrosequencing. So we just suck at all of the DNA, and we sequence it, and we can identify the microbes this way. And this is really, really small, and you can't read it, and that's okay, because the only point I wanted to say is these samples are actually very diverse. There's a lot of bugs in here, and in fact, a lot of these samples don't share families between the samples, even though these are cores with depth, and just centimeters away from each other, you'll find samples that don't share any bacterial family. So these are very, very diverse samples. Just to kind of draw your attention to some sample or some, I guess, bacteria that I'm finding. So I have cyanobacteria, some methylotrophs, Dichloromonas, which is perchlorate reducing, pelagibacteria, that's a common, fairly ubiquitous marine bacterium, tons of actinobacteria. I've replicated this particular sample, and it turns out that I think this just happened to be the 5 grams of soil I sampled just happened to have a lot of spirochetes. So they're extremely heterogeneous soils. Oh, wow. All right, all right, there's bacteria in there. There's also archaea in there. This is really special because the archaea were not really known to be in the upper dry valleys until very recently. So this is really interesting. They're not that similar to what we know in GenBank already. But I did do some in situ work in the dry valleys. I tried to measure any gas flux coming off of the permafrost, and it turns out there's not a lot of gas flux to measure. So in the lab, one thing that we can do is we can feed our soil samples some radioactive food, and then measure the radioactive carbon dioxide that comes off of those samples. And this is what that data looks like. This is about a year-long experiment. And so if I'm looking at University Valley, there's a shallow ice table and the mid-ice table. The ice table, I just mean that how close it is to the surface that I was showing in that diagram earlier. And what we see is that at 5 degrees Celsius, we do get some mineralization in this shallow ice table core. But at sub-zero temperatures, we don't actually see anything. And my control samples are coastal Antarctic sample, which I thought I'd have better luck and to just make sure my experiment was working. So in the case of the shallow ice table core at 5 degrees Celsius, we do see mineralization. It's about after 150 days. This is a temperature that these bugs would never see at their site, and certainly would not see it for 150 consecutive days. If you were to ask, are these results because, let's make things a little bit more favorable for any potential microbes in there. Let's throw some nutrients in there. Let's make it a little bit warmer. At 10 degrees Celsius and adding some yeast extract, that actually has no effect whatsoever on my University Valley soils. But with my coastal Antarctic soils, it actually increases the mineralization rate by about 10 times. So is anything actually alive in University Valley? In the permafrost, they're not happy. And if anything is alive, I'm not able to detect it using the methods that I'm using right now. But it turns out that there is life here, it's just hidden. And so if you look at these valley walls, and it's a tough job, but someone gets to do it. And if you go up those sandstone walls and you just whack a rock, you can actually, almost any rock there, you can see microbial life, and you can see evidence. And it's unfortunate that you can't see how green this is. It looks great on my laptop. But you'll see some photosynthetic cryptoendolithic communities. And they're actually fairly ubiquitous in the dry valleys and at the site. So this is one in the lab, and you can stick them under a microscope, and you can see the microorganisms. There's algae, and there's cyanobacteria, and there's plenty of fungi. I'm pretty much just starting the activity work on this now. I have some cultures in the lab. They are growing at 0 degrees Celsius. I'm having a lot better luck with the cryptoendoliths than I did with the permafrost isolates. So I've only got 6 permafrost isolates. I have over 40 isolates from the cryptoendoliths already today. And so it's a lot to work with, but they're a lot easier to work with. And so why is that? Well, the cryptoendoliths, besides having access to the sun, as an energy source, it's also a little bit of a warmer and wetter place. So these rocks actually get heated up by the sun almost 20 degrees warmer than the air temperatures. But it also, it's very porous in there, and so that provides a surface area for any available water in the atmosphere to condense upon. So it's a little bit wetter, and it's a little bit warmer, and this provides a place for bacteria to thrive. So is anything alive there? Not to be cliche, but thanks.
Thanks, Jackie. And we have maybe time for one question. Does anyone have a question for Jackie? Okay.
So you fed them yeast extract and they didn't grow, and you're giving them acetate and you're seeing low remineralization rates. Do you think that's just an effect of they're not happy with what you're feeding? Maybe try like maybe 14 glucose to see if a different substrate gives you a better yield?
I can try 14 glucose. This actually took me a really long time to do, and it's worth doing. But to be honest, I don't think it's an issue of the carbon source. I think there's either nothing there to actively metabolize. I just think or they're taking a really long time to recover. The one that did show mineralization at 5 degrees, and I wish I had more time to kind of dwell on that image. There was a really long lag period before it started mineralizing. Over 100 days.
Ah.
Yeah.
So thanks, Jackie.